So we're going to talk about how we have horizontal or lateral transfer of DNA. So transformation is the first one. So it's from cell to cell. It was discovered in 1928 by Frederick Griffith. And I have the next slide explains how he figured it out. So basically, two types of pneumococci. Okay, so so there it is. It has a capsule. And if it had a capsule, he called it smooth. Okay, and this one was pathogenic. Okay, so that means it actually killed the animals. So if he gave them live pneumococci that was smooth with the capsule, gave them mouse that, it got pneumonia and it died. Okay, there's another type of the organism that didn't have a capsule. Okay, so no capsule. He called it rough, okay, because when you looked at the colony on the plate, this colony over here was really smooth, and this colony was rough. Okay, so he took rough, that was live, and he put it in, and the mouse lived. So the conclusion is that there's something about the capsule that made it more pathogenic. So then he heated up the organism with the capsule. So he killed it so that the organism wasn't alive, and he put it into the mouse, and the mouse survived. Okay, so he didn't want to stop there. So what he did next is he killed smooth pneumococci, okay, that has the capsule, so it's dead, okay, and he mixed it with the live non-virulent one in like a test tube or something, and then he injected into the mouse. Okay, the mouse actually died, right? So nobody was expecting the mouse to die, because essentially he was doing this, right, and this into there. So you would think, or not this, sorry, this. Oh, shoot, okay. This and this. He was putting those two together. So you would think that the little mouse would live. But, it would probably be more than one mouse. Okay, so after the mouse died, they did Koch's postulates, right? And they pulled out capsulated pneumococci and uncapsulated pneumococci. So what they determined is that some DNA from the dead one, which was the gene for forming the capsule, got transferred to the rough. And then we created these capsulated ones again. Okay, so this is where transformation occurs. So the idea is that if there's free DNA from an organism, from like a different bacteria, from one cell, it can be taken up by another bacteria. Okay, so these other bacteria, they have to be called competent. So in order to be competent, when you're competent, you're able to take up DNA. Okay, so essentially the naked DNA from the smooth ones being killed was floating around in the solution with the rough ones. And the rough cells were actually competent, and the DNA went into them, and it became integrated and became part of the chromosome. So this organism here, okay, this one, which would be this one, was actually transformed. Hi. Okay, so that's transformation. Okay, now transduction we've already talked about. So transduction is when we've talked about bacteriophages. So remember when we had the cell, we had the bacteriophage, okay, and we had DNA in here, okay, and the DNA got injected into this, okay, and it became part of, okay, so here's your chromosome, DNA becomes part of it. Okay, so that's transduction. So transduction is virus to bacterial cell. That's how it's moved. So Liederberg and Zinder figured it out in 1952 using salmonella. There's two ways that the information can be transduced. It could be generalized, where it goes into any part of the chromosome, and then there are some that are specialized and they go into specific regions of the chromosome. Okay, so why do we care? Why is this significant? Okay, so it is a way to move genetic material around. People are, like evolutionary biologists, are always wondering. So if you remember transcription and translation, eukaryotes, we have information that's being expressed, okay, and then we have information in our genome that's not being expressed. So they always wonder why do we have this not expressed information? Why do we have this? So some people propose that it could have been a virus infection, that it went into the host and it's just always been there. Okay, this also helps explain the viral origin of cancer. So if a virus gets in and it puts a piece of DNA into a chromosome, it can cause uncontrollable cell division. Okay, conjugation we've also talked about. So conjugation is when we form the conjugation pilus. Okay, and then information can be transferred along that pilus to the other cell. Okay, so in order to donate DNA, the cell has to have this thing called an F plasmid, F plus plasmid. The F plus plasmid actually contains the information to form a pilus. And that the stuff to form the pilus can be transferred. Okay, so significance of conjugation, it contributes to genetic diversity. It's useful if we're trying to map a chromosome. We can move plasmids around, and we're going to talk about that for biotech stuff in a second. And it's a way that antibiotic resistance can be transferred. Okay, now, biotech stuff. Okay, so so if you are working as a healthcare provider, okay, I feel, and it is becoming more common, that we're using recombinant DNA, recombinant DNA techniques, okay, to create drugs, to create medications, and to treat patients. Okay, so I want you to understand how this process works in case somebody asks you. Okay, so here's the idea. So let's say we want to talk about hemophilia and factor VIII. Okay, so blood clotting is this complex cascade of events. And there are all of these proteins that are called factors that contribute to the blood clot being formed. People that are hemophiliacs can be missing different factors. Okay, an example of one of the factors is factor VIII. Okay, so what they would do is they would take so this is a healthy cell, and they would look for the gene of interest. Okay, so they're looking for the gene that's going to code for factor VIII, because factor VIII is protein. Okay, so they're going to collect the DNA, they're going to cut it with these restriction enzymes. So restriction enzymes just cleave DNA, and they're going to find the gene for factor VIII. Okay, and then what they want to do is they want to be able to put it into another organism. Okay, so they can cut it out, okay, and then they can paste it into a plasmid that we can buy. Okay, so this plasmid will then, it's called a cloning vector, is the plasmid, and you can stick this piece of DNA into the plasmid and ligate it using DNA ligase. Okay, so you end up with the plasmid that's recombinant, so it's got DNA from two different sources. Okay, the other thing that's on this plasmid is it has an R gene. Okay, so remember, those are genes that code for antibiotic resistance. Okay, so let's pretend this one codes for ampicillin resistance. Okay, now what they're going to do, or what you can do even, is you can take this plasmid, okay, and we're going to put it into a chromosome, or not chromosome, we're going to put it into an organism. So we're going to put it into E. coli. Okay, so we're going to take young E. coli, okay, and to make it competent, it's got to be at log phase. Okay, so that's what competent means. It can take up DNA, so they have to be young. Okay, so you take E. coli, okay, and we're going to add the plasmid to the cells. Okay, and then we can treat them with chemicals, or we can shock them with a jolt of electricity. Okay, and we can get the plasmid into the cell. Okay, now the plasmid is not going to go into all of the cells. We're not going to get 100%. So this is called transformation. We're not going to get 100% transformation. It just doesn't happen. It's not that easy to do. Okay, so some of your cells will have plasmid and some of them won't. So how are you going to tell which ones have it and which ones don't? So the plasmid has antibiotic resistance in it. So what you do is you take all of these and you put it on media that has AMP. Okay, so if they don't have the plasmid, they're not going to grow. And if they do have the plasmid, they're going to grow. Okay, so then what you can do is you can grow these up, okay, grow a whole bunch of them, and you can get the plasmid to make the factor 8. You can make these E. coli cells make factor 8. And then you can harvest it and you can purify it and you can use it to put into people. Okay, this is a chart from your book. Okay, so this is probably not everything, but this is a list of things that have been created using biotechnology. So some examples of stuff would be human growth hormone, EPO, blood clotting factor.